an RNA molecule is created, it's usually termed a pre-RNA molecule. So it can be a pre-mRNA, a pre-tRNA, pre-rRNA, and all it means is it's not the mature form of itself yet. So there has to be some kind of processing that takes it from being a pre-transcript into a mature one. And we're going to examine some of those. Okay, and this is an overview of modifications that can occur. So the first we're going to look at is cleavage. So here we are looking at a rRNA transcript. So when it is first transcribed from the rRNA gene in the genome, it creates this primary transcript. It's 45 S's, and that's how it's being measured. This 45S transcript has to be cleaved to produce the mature 18S, 5.8S, and 28S that go on and help create the ribosome. So you have endo and exonucleases that come in and clip out the parts that are necessary to form the mature. Okay, so our RNA molecules have this primary transcript. It's going to be cleaved by endo and exonucleases that leave you with the mature transcripts that's necessary. So this is the processing of our RNA molecules. I'll also note that this occurs in the nucleolus. Why in the nucleolus? Because that's where ribosomes are made. So the nucleolus of the nucleus is usually where you find our RNA genes. The tRNA is also processed through cleavage. So you have a precursor tRNA molecule that's created, and that's what we see here on the screen. And so what happens is first, it's cleaved by RNase P, which is an endonuclease. Okay, and so that's going to produce one end of the mature. Then you have another endonuclease that removes a segment from the 3'. Then you have an endonuclease that cuts here. Then you have an exonuclease, RNase D, that cuts here, leaving this ACC, which is indicative of all mature tRNA molecules. And then there's some base modification that occurs, and we'll talk about why that's important. And that also helps us to recognize which tRNA molecule is which one, and to correctly put the amino acid that belongs with it. Now, some of this is created. So RNase P is a ribozyme. So what is a ribozyme? So automatically, when looking at this word, it resembles enzyme. So that should tell you it's an RNA molecule that has catalytic activity, because we know under normal circumstances, enzymes are made of what? I'll leave that there because I'm quite confident that we all know the answer to that. And so this is another way of looking at the tRNA processing. So the darker green is what becomes the mature, and the light green and the dark green is the precursor. And we can see how pieces are being cut off along the way. And then the dots represent modifications that occur. So for some mRNA molecules, so remember mRNA molecules transcribed by RNA polymerase II, and they're going to go on to make our proteins. So we talked about how they have these introns. So we have to splice, cap, and tell to create a mature. So in the pre-mRNA, you have exons and introns. Exons are the expressed or the coding regions, and then you have introns in between them, which are these intervening sequences or non-coding regions that have to be removed, and then the exons have to be pasted together or spliced. So there are different mechanisms that this can occur with. So there's group 1 and group 2, which are self-splicing mechanisms. And then there's splicing with the spliceosome, which mRNA uses, and we'll go into more detail with that. So if they are splicing, so group 1, this is found in some rRNA genes in some simple eukaryotes, some protein coding genes, some tRNA and rRNA in mitochondria, and in some chloroplasts. Group 2 splicing is found in some protein encoding mRNAs, some tRNA, rRNA, also in mitochondria and chloroplasts. And then we know that most protein coding genes that make mRNA in eukaryotes do spliceosomes. So how does group 1 and group 2 work? In group 1 splicing, so this is also examples of ribozymes. Okay, these are self-splicing. So this guanosine breaks the bond between the exon and the intron. So this guanosine is active and it's able to catalyze the breakage of this exon from this enzyme. We see that? And then this is going to go in and attach to the other exon, ticking off that intron. This is group 1. With group 2, it's catalyzed by an adenine. So this adenine is able to break the bond between exon 1 and intron 1, and then exon 1 joins to exon 2. I'll also note here that in cells, this process is usually sped up by maturases or maturases, depending on how you pronounce it. Okay, so these are enzymes that enhance the rate of self-splicing. Now let's talk about splicing with the spliceosome. So the first thing to note is that it requires 3 sequences. So here we go again with our consensus sequences. So between the end of an exon and the beginning of an intron is the 5' splice site. At the end of the intron and beginning of the next exon is the 3' splice site. And then in the middle, you have this branch site. Okay, and this A here is important in that branch. These are consensus sequences. So A or C, G, GU, a purine. Any amino acid, a pyrimidine. Okay, so that's, remember how we read this. All right, so how do we get this? We have a spliceosome. So this requires SNRMPs, or what we're going to call SNRPs. So these are small nuclear riboproteins. Riboproteins, a mix between ribosomes and proteins. Are we okay with that idea? And so what happens is together, these SNRPs have these small nuclear RNAs, so these snRNAs, and then proteins, and they make small nuclear riboproteins. And they are called U's. So U1 is going to represent or going to bind to this 5' splice site. U2 binds to the 3'. U4 is binding to the branch site, and they start to move together. Collectively, they are the spliceosome. So like most things we talked about, it's different subunits. So what does the spliceosome do? It binds to introns, recognizes this intron-exon boundary. It holds it in the correct configuration so it can be spliced, and then it catalyzes the reaction that takes that intron out and puts those exons together. Okay? So U1 is binding to the 5', U2 is binding to the splice site. They're going to bring it themselves together and form this lariat, and that then U5 and 6 are at the ends to do the bringing together of exons 1 and 2. This is the lariat. So this is like this little loop-like structure. This is indicative of introns that are removed. They form a lariat. This is just going through the process again in a different way. So if you prefer to look at it this way, then here's a picture for you. Okay, and the same thing again. It's several different things are involved. You form the lariat to take out each intron. What's also important is this idea of alternative splicing. So what does that mean? You can take the same gene and create different proteins from it. And one of those ways is by this alternative splicing. So if we look here, we have our DNA, we make our pre-mRNA. Now how we splice it, we can have a mature mRNA that has 3 exons, or we can have one that has 2. And now these are going to make 2 different proteins with different functions. They could be similar, but they are still different. So if we look at it in a different way, this red here represents constitutive exons. These are ones that are going to always be there, always there. The green are the alternative ones. So they could be in or out depending on what we're talking about. So here we have alpha tropomyosin. And this is going to exist differently in smooth versus striated muscle cells. So remember, your DNA is the same, but because these two cells need different versions of tropomyosin, they can alternatively splice it. And you'll see in striated, it's longer. It has 3, whereas here they have 2. This has 11 and 12, this has 14. So this is alternative splicing that's giving you 2 different function in tropomyosins depending on your tissue type. So these are some other ways that alternative splicing can be included. So the cassette exon included or not, include one and not the other, keep the intron in, have alternative 5', so this won't be this big, that won't be that big, alternative promoters, so this starts here or this one is here, and then alternative splicing and polyadenylation. So how do they do that? So there are splicing repressors and enhancers. So if you repress a site, then you're hiding this 3', forcing the spliceosome to recognize the next one. If you're enhancing it, you're saying, hey, make sure you get this one here, okay? So these are ways that alternative splicing can be achieved. Now, the next thing with mRNA processing is capping. So at the front of the sequence, we have to add this methylated cap, and it's a guanosine cap. And if you look here, you see the G added. So this is often called a 7-methyl G cap. Okay? So, and this is like a reverse orientation. We cut, invert it, and put it back. And you have this 7-methyl cap. It has several functions, very important, and you have to have it. One is it's going to help us to get out of the nucleus. So remember, the nucleus is like Fort Knox. Only things that are allowed can go in and out of it. So this is going to help us take this mRNA out, because if it stays in the nucleus, we can't use it. It's going to help us start translation. So this cap is recognized by initiation factors in translation that helps it to get to the ribosome and be able to be translated. And then it also helps in splicing. Now, these used to be called post-transcriptional modifications, but we now know that they are co-transcriptional modifications. The other thing is we have to add a tail. So remember the polyadenylation tail, a bunch of adenines are added to the end. So there's a consensus sequence that tells us, okay, 11 to 30 nucleotides downstream of that is a cleavage site. Cut there, add the poly A tail. There's alternative polyadenylation though. So depending on where you cleave, you can have a longer or shorter mRNA. So here we have one that's exons 1, 2, 3, and 4. Here we have 1, 2, 3, 5, and 6. So this is polyadenylation and alternative splicing. And the last modification is RNA editing. So with RNA editing, we can change the sequence of an RNA molecule. So we have these guide mRNAs that are complementary to our mRNA sequence, and it serves as a template that allows us to add nucleotides, remove nucleotides, or alter nucleotides that are there. Collectively, the importance of alternative splicing, alternative polyadenylation, and RNA editing is to allow us to use the same DNA molecule and make different proteins from it. Okay, now we have talked about how transcription occurs in bacteria and eukaryotes, and I am confident that you can fill in this chart accordingly.